So now I launched it one more time. Fine. So we discovered that our cells have some interesting property, and this property is that they pull. They almost exert pulling forces on the environment. And I also mentioned that nobody knows exactly why they do it, but still there is a nice and beautiful model hypothesis formulated by Donald Ingber, and I will now introduce it, and I think you enjoy it, but you can agree or maybe you will not agree with this idea. We can also maybe talk about this. I can make an examination question out of it so that we can exchange our opinion, what you think about this idea. By the way, it is not only those collagen gel experiments where we see that cells are pulling. Today, in the finalizing part, in the last part of today's class, we'll talk about different methods how to measure cellular forces, and all of them, in fact, bring same results. So every time we measure forces applied by the cells, we will always find a pulling component in them. So this is one of the methods used nowadays. So traction forces measurement, and also in this case, the cells are cultured on substrate containing such elements, such sticks, and if you just track their position, you will always find that the cell always tries to deform them in this pulling way. So if I'm not wrong, the concept of tensegrity was first introduced by Albrecht Bühler, who mentioned that sometimes if you want to build something, you cannot rely on gravity. Let me explain what he meant. If you are building a house, you usually put some bricks and you connect them, of course, by some concrete. Sorry, there was a question. Are we talking about adherent cells or free movable cells? Adherent cells, yeah, you're right. So free movable cells, well, depending on what you call free movable cells. If we're talking about like moving cancer cells or leukocytes, they also do produce a lot of pulling forces. But if we're talking about red blood cells, they do not. Yep. So that from this point of view. In other words, every cell that has connections that establish mechanical connections to the environment, those connections are always somehow stabilized by pulling. So the cell never, if it catches something, it never just catches it. It also always also pulls it. Okay, let me come back to the ideal building. And so you know that most of buildings we have here are stabilized by a gravitational force, and we use quite heavy building blocks. And also, if you check the structure of the Kölner Dome, of the Cologne Cathedral, you also find out that they use special tweaks to apply gravitational forces in a very stabilizing way. So they have lots of contraforces and external walls that support this very high building. But quite soon, many people discovered that if you are in open space, maybe orbiting the Earth, then you cannot rely on gravity because it cannot stabilize the structure. So you need another way to to keep your parts together. And interestingly, the same problem is encountered by very small organisms. The reason is that as we're getting smaller and smaller and smaller, you will see that some forces that dominate on our scale, they will almost disappear on small scale. For example, gravity will not contribute anymore. If you ask a bacterium about gravity, if bacterium, if it could speak, would tell you, no idea, I never heard about gravity, because what bacteria or even insects deal with are mostly van der Waals forces, or maybe surface tension forces, or viscosity, because on the level of bacteria, water is so viscous, it is the region of such low Reynolds numbers that they mostly deal with viscosity only. So this is the main headache actually for them. And the same is true for our cells. So they do not feel gravity, but they need to have something to keep their parts together. And this something can be tension. So there are some comments from Lorena. So about traction force measurement. Lorena, I will tell about these methods in the next part, okay? So actually, if you still have questions afterwards, of course, I will answer. Yeah, this is it's called not fraction, but traction, I think, traction, okay? Maybe just an example for you. Think about what happens now I'm sitting on the second floor, and if I take an ant or insect and I throw it from my window, so I think for most insects nothing will happen, right? They just fall down, they say, oh, okay, and they will just crawl further. But what if I jump from my window? It would be not so nice. I may break my bones, but if I take even a heavier creature like an elephant, so an elephant will definitely break all its bones if I throw an elephant from my window. So the larger the object, the more dominating will be the gravity force, right? So as we are small, we need to apply more other forces than gravity, and one of them is tension. And indeed, using tension is one of the favorite methods applied by living creatures. Some of you with background in medicine or physiotherapy, you also know that human body is sometimes considered from the point of view of tensegrity as a system consisting of supporting elements that withstand mechanical compression, and those are bones, of course, and cartilage, of course. And we have also some tensile resistance components in our body, and those include different ligaments and fascia. They actually deal mostly with the tensile load systems. So also human body is a nice example where different sorts of forces, tensile and compressive forces, are are managed in a very coordinated way. Also, our spinal cord can be seen as a wonderful biomechanical concept that connects certain compressile elements and also tensile elements. And the same would be true for on microscopic scale, as you check the connective tissue network mesh that fills the space between our organs. Again, you will find some interesting combination of tensile and compressive forces. And now we are down to the cellular level scale, and what we see here is this beautiful triangular architecture, which is again, from the architectural point of view, is a way how to distribute forces in a nice way so that the tensile force never will be too high to rupture the cell, and also compressive forces would be compensated in a wonderful way. So again and again, in all living creatures, we see actually same idea of unifying tensile and compressive elements together. And slowly we are now approaching the tensegrity concept, which I will define a couple of slides later. If we also examine some objects from our environment, we will also notice that many of them are stabilized by tension. So if you remove tension from the spider web, it can collapse, right? So if you cut the ropes, it will collapse quite quickly, and the spider will be quite unhappy, right? And yeah. The same is true for if you use a tent, and I think some of you have experience in living in such a tent. You also know that it is important to keep it very at high tension, because otherwise it will collapse and it will be quite unpleasant. Now I'm just thinking about as I was student, that was 1992, we had some practicals in mountains. So 40 days long, we needed to live outside in mountains, far from civilization. So we lived 40 days long in such tents, and there was a lot of fun in the night to cut all the ropes, and so that the person will be entrapped in such a tent. So for this person, that was not such much fun, of course, but that was actually quite obvious how important those tension in the ropes were for the stability. Well, maybe foam is in this case not such a nice example, because I think foam is partially stabilized by pressure, right? So it's more pressure stabilized structure. But spider web and tent, I think they are good examples of structures that rely on tension, and if you remove tension, they collapse. So, and now I want slowly to bring you to the idea that maybe also our cells, and here we see our monocyte, also many of our cells are probably relying on the same idea. So that's why they apply tension, because without tension, they will be not stable. They will collapse mechanically. What I mean with collapse, I mean that such a cell will collapse to a spherical shape, and that will be that's it, because it will be due to surface tension, it will just have surface shape and cannot have any other shape. But you know that in our body, cells have very different shapes, and it would be actually bad if all our cells in a body would be spherical. So now we are reaching the definition of the tensegrity architecture, and again I have to mention some famous names, some Nobel Prize winners, like Buckminster Fullerene. And later it occurred that such tensegrity-like principle is visible on various structures, including buildings, human body, polymers, and so on. And so that's now the main idea of the tensegrity principle. As the word itself suggests, it is a combination of two words, tension and integrity. So if you make a hybrid of these two words, so you get a tensegrity. In other words, integrity which is appearing due to tension or tensional integrity. And the main principle that which you please have to notice, and that's why I labeled it in red, is that in a tensegrity structure, mostly tension is continuous and compression is local. It might sound puzzling for you. That's why let me explain it a bit. And also I want to say that not all tensegrity-based structures, they are perfectly applying this principle. It's not perfect, it's just an ideal principle, but of course, if we check cells and human body and many other structures, this is not exactly this principle, but very close to it. And the idea is that we have a combination of two elements. So we have struts, and those are elements supporting compression. And we have filaments that logically will take care about tension. Both these structures are working together in such a way that struts, they are positioned in right positions to address the local load, compressive load, whereas filaments are rather spread or distributed more uniformly, and we have a network of filaments, and this will enable them to distribute tensile stress between many elements. In other words, if I'm talking about compression, this strut will deal with this compression, local compression alone. It is a kind of egoistic approach. So it is like, yeah, it's a lonely supportive element, maybe unhappy, but the tension is shared in a whole group. That means this rope, which I'm showing now, is not the lonely rope, but it just goes further and then further and further and more and more and more. And so that the tensile stress and actually is going through the whole structure. So it's shared by the element, and it is distributed between this element such a way that now if I apply high tensile stress, it will not rupture, but it will be uniformly shared between many elements. So this is just to explain a little bit this concept. Tension is continuous here, is distributed, and compression is rather local. So you don't need to write much concerning your notes. I guess the main idea is here, and if you understood the main principle, The rest will be easy for you. Now I will just show some more examples so that it will be impregnated into your mind. And if I wake you at night and ask what the concept of tensegrity is, you will know it definitely. So these are some molecular simulations, computer simulations on tensegrity-based architectures applied for the cells. And now I'm referring to the paper of Donald Ingber, whom I mentioned before. He's a scientist who first introduced the tensegrity concept into cell biology. And you can find a dozen of papers from him. I think he started 1990 something publishing papers, and there are many papers published, and all of them, or many of them, devoted to this idea. And here's one more computer simulation where they try to understand how the force can be, meaningfully distributed, how tensile and compressive elements can nicely share the forces to make the cellular stability maximal. And those ideas are also used not only in cell biology, but also in architecture. In the Netherlands, there is such a high tower that is totally built by using tensegrity principles. Please notice that the single compression supporting elements are not connected to each other directly. So there is no glue between them. There are no screws, there are no bolts between them. The only way for them to stay together is to be connected by the ropes, so through the ropes. And if you cut the ropes, the whole structure will collapse, in fact. So it is ropes that make this structure unified. You remove tension from the structure and it will collapse. Same principle has been applied also in some buildings in China. So this is one photo I made in our trip in Shanghai. And again, this was a good example of this tensegrity architecture. They used tensile force to stabilize these buildings, so like tent-like principle. As for the cells, we also know that there are lots of candidates that can be mentioned as tensegrity elements. So we've learned microtubules, we've learned intermediate filaments and microfilaments, and I think it is very clear for us who of them will be the tensile supporting elements in our cellular tensegrity model. Do you know, don't you? So I've got such a choice: microtubules, intermediate filaments, microfilaments, right? Not exactly right. Okay, so there are a couple of answers. The right answer is intermediate filaments, actually, because they are the strongest tensile elements. They, as we saw today, they have the largest tensile rupture energy, and they are mostly suitable as tensile resistant elements. Actin, not so good. It can work, actually, indeed, and microtubules would be the worst. So if I put it as a priority list, intermediate filaments, actin filaments, microtubules. What could be then the compression supporting element inside the cell? Well, that will be hard because microtubules are the stiffest elements, but even microtubules are not too strong. So they could partially take a function of bones in our body, but compared to bones, they are still quite weak. And they can bend easily if you try to load them. And that's why the cell needs to use some external environment which will allow it to avoid the compression forces inside. Let me explain it this way. Instead of producing compressive forces inside, the cells produce tensile forces outside. I think you all understand what I mean. If I will make a drawing still. So here, let's consider some force, and let's put this force in this direction. So seeing from the cell, this is a pushing force, this is a compressive force, and this force can be actually supported or opposed by reaction force from the cell, or it can be opposed by reaction force which will be applied here from the environment, from the outside. And this is the concept that cells have to use because they are lacking very strong compressile elements inside. The microtubules, okay, but not so good. Well, actually, you need to have some connection between your elements. And here are some examples. I don't want to stress this figure much. It's just for your information. So some of you ask just me to make it very clear which will be asked, which will be not asked in exam. So those details, they will be not asked, right? But I think it's also a little bit interesting and funny for you to see the connections. So here we see the red blood cells, part of the red blood cell membrane. And in the red blood cell membrane, we see Band3 protein, which is a channel, in fact, transporter. And this transporter is anchored to the extended two-dimensional cytoskeletal network, which helps red blood cell to withstand mechanical stress. So also red blood cells, although they are not typical cells, even these cells, they show certain elements of tensegrity principles so that they also have some distribution between compression and tension. They actually don't need to deal with compression too much because they are greatly stabilized by stiffness of their cytoskeleton, and it is enough. And secondly, they are so densely filled with hemoglobin that they don't need to care about compressive forces. They are like tiny sacks, tiny bags filled with hemoglobin, and that's why for them it's a little bit different. A couple of more pictures. What you see here is on the left side is the cell which is not subjected by stress. So this is a resting cell. And on the right side, you see cell that is loaded, mechanically loaded. Do you see the difference, right? So this is resting. This is loaded. So meanwhile, I see there was a question from Ina. Is the cellular tensegrity directly affected by changes in temperature? Well, interesting question. Honestly, I can just deduce something, but it's so complex, you know, if there are so many players starting from Prp channels there. I don't know, honestly. Good question, but maybe there is a paper about this. We need to check. So interesting aspect. Let me just admit I don't know. Well, what I can tell you actually concerning temperature is that the cells usually shrink if they have too cold environment and also if they have too warm environment. So they react in a very complex way. So you heat the cells and they shrink, and you cool down, they shrink as well. But again, it depends on the cellular type. Yeah, too complicated, sorry. Again, if I'm thinking about endothelial cells and smooth muscle cells, so they react again very differently. And okay, sorry. Let us think about this maybe some other day. Well, and now I want you to see behind this photo the tensegrity picture of it. And what we see here are unloaded green tensegrity elements, so struts and tensile elements. And this is what happens if you apply mechanical load to this structure. By the way, I have a toy which I need to find now, just a moment. So, you know, in my office, I always have some toys because if it's too boring, I need to play with it. So, look, this is an interesting and good example of a tensegrity structure. So, it's not very perfect, but very close. They have separate, so let me put it like this. So, there are separate struts, there are separate compressive elements, and they're all connected to the same rope. And the rope doesn't need to be rubber-like, it is just a normal rope, but look what happens. If I now try to pull this structure, it pulls perfectly, it reacts very elastically. It says, fine, no problem, you're pulling quite strongly, but we do not rupture, we just change our shape. And I'll say, okay, let me destroy you by using some other way. Let me just now push you. And again, they say, fine, no problem. We can withstand your forces, so we can also deform in an appropriate way. And whatever I do with this structure, it is made by rigid elements, in fact, but it reacts as an elastic entity. And to me, this is a nice example of a tensegrity-like architecture. So now, I hope I tried to explain some ideas, current ideas or state of art, what we know about forces, force distribution in the cell, and how the mechanical loads might be distributed. I tried to make it more or less compact, and actually the only thing you really need for the examination is just to have an idea what is the main message of the tensegrity concept. and what are possible kind of proofs or evidences that such mechanisms could work in a cell. So I like this idea, and for example, my former boss, Professor Artman, he didn't like it, so we spent many hours in discussions, what was interesting. So not necessarily that Tensegrity is the only truth, you know, but at least I think it's quite beautiful, and well, why not? Fine. Now, before the break, I want us just to quickly overfly the aspects related to cellular motility, because there will be lots of things that we've spoken about, and there will be lots of things that we don't need to know in detail, okay? So let me now, let us go through, it will take 10 to 15 minutes. Let us overfly the aspects, what we know, what we learned about cellular motility, cellular motors, and then we will finish this section. Okay, so please, I require just 5 to 10 minutes your concentration, which is not, you don't need to be very concentrated. That's actually fine. Okay? So first of all, I hope that the video will work. If not, then so these are moving cells. So 3 line, we have also in our lab. These are mouse cells. These are transformed mouse cells, so they are immortal. So an immortal cell culture we call cell line. And what we see are mouse fibroblasts. And you see that they are moving quite actively, and it's really amazing how you know how they move. So there are so many interesting events. So you also saw some dividing cells. This image has been obtained with the help of phase contrast microscopy, so without staining. And what we see are really living cells, and the cells not affected by any regions. So we've already spoken about different types of cellular motion. Let me remind you, we talked about actin polymerization. We also spoken about Brownian motors. The actoclampene motor I kind of canceled because I said it's not, seems to be not true. I also introduced the gel-sol mechanism, this osmotic mechanism of motion. There could be mechanisms related to vesicle transport. I will explain it now. And myosin, we also discussed in this course already. Let me now very briefly say what I mean with vesicle transport. Just a moment. By the way, I'm quite proud because it was my idea. And a couple of years ago, they said so in some papers they mentioned they saw it. So that's why I'm, of course, very pleased that my speculation worked there. Vesicle transport mechanism can be seen like this. So look, what if we have cell, and the cell can take part of its membrane from this part, so it will form some invagination here. It will make a so it's not liposome, like vesicle, membrane vesicle. Take it from membrane due to engulfment, and will transport this vesicle to this part of this cell, and this vesicle will be integrated into the membrane. You understand what I mean, right? So you are taking membrane from this place, and you are bringing it to this place. Now just imagine what happens here. You are removing membrane or making its area less from the left side, and you're increasing the membrane area on the right side. And now just imagine you repeat it many times. What do you see? You will see that you have more and more membrane from this side, and your cell will move in which direction? Think about this. So it should move to the right. Yeah? So if you transport your vesicles and you play with membrane, you can also imagine such type of motion. I'm proud because I just played with some possible hypothetical mechanisms of cell locomotion motion. And yes, thank you for your answer. I just suggested this. That was not published, but I spoken with my colleagues, and I just said, oh, that would be a nice, funny way to propagate, actually, to move. And then I saw that there were publications where they mentioned that they saw this way used by immune cells in some cases. So, of course, I was very happy that maybe my idea was true. What I think a bit pity that we haven't published it by that time, and now I cannot really claim, but it's okay, no problem. So let me come back to this slide. So this we did, discussed, and we talked. What if I ask you to give your own definition? What is molecular motor? So what would you say? What is a molecular motor? Imagine you are a Wikipedia and somebody wants you to make a definition. So what if you are Wikipedia for some person? So let us try. Molecular motor is, so since it's a molecular motor, right? So we say it is a molecule, right? And in many cases, we can even say it is a protein, which is capable of converting chemical energy into mechanical energy. So I think it's not bad. So not perfect, but I like it. It is a molecule that is capable of conversion of chemical energy into mechanical energy. Why chemical energy? Because it could be energy of gradients, it could be energy of ATP, it could be some other energy, but in all cases we can say, okay, this is sort of chemical or maybe electrochemical energy. And mechanical, because it could be indeed very different. So it could be a rotational energy, or it could be translational motion, right? So that's why I think the definition is not bad. So in our written exam last year, there was such an example. We needed to write such a definition, and I think that wasn't too difficult. So we're talking about actin-based motion, so it could be done by many ways. There are also, of course, some ways realized for microtubules. And I know that you're tight, just a couple of minutes. There are actually many interesting options realized in the cells. For example, the cytoplasm is making this circular motion, this water streaming, which probably occurs due to actin activity. We also spoken about some cellular motion. I already introduced it, but let me briefly repeat. Due to actin polymerization, it is pushing membranes so that the filopodia or lamellipodia is formed. Then attachment follows, so you are just use your hands and it grasps the substrate. Then the contraction phase is coming, so the cell pulls its body towards the attachment site. And then the release occurs so that the cell will detach from the behind substrate. And then the whole cycle is repeated again. So basically, this is the cycle of cellular propagation for many cells, including fibroblasts. To me, this whole scheme resembles mountain climbing, rock climbing. Do you agree? Maybe. So to me, it's very similar. So imagine you are this rock climber, and you are sitting in the middle of the rock, attached to the rock, so not to fall down. Then you just make next attachment point, put a nail there, attach a rope, then you pull your body upwards, then you release the ropes. You just settle in a new position. Then you again extend your hand, make a nail, attach to the rock, and you pull yourself upwards, so you are like propagating. And this is the way how many cells they are moving. This is rock climbing mechanism. So also shown here in some pictures. We don't need details. It's just some fine things that you can expect by yourself. So we also spoken about there could be different polymerization approaches. Dynamine we haven't spoken about. This is too much. We also learned about cytoskeletal motors. I hope you remember the names: myosines, kinesins, dyneins. And there are also nucleic acid motors, just to know. And there are also synthetic molecular motors, which we don't need to study in this course. And these are our familiar things: myosin, kinesin, dynein, that are associated to microtubules. Here are some portraits of them. This is our Brownian ratchet mechanism, or also sliding mechanism that we discussed before, but you don't have it in your script. Bacteria use another mechanism, which is in fact based on rotation. It's kind of very funny rotor mechanisms shown here. As researchers first saw it on electronic photograph, they couldn't believe it because it's so weird. They said, no, nature did not invent a wheel. but in reality, nature did invent wheel-like structures, and this is an example of that. So if somebody tells you that it was humans who invented the wheels and gears, it's not true. So bacteria invented wheel-like mechanics. Well, and that's it. So you notice that in the end, I just made it quite quickly. The signal for you is that it's probably not so important, and you just got some pictures. So, okay, there are different kinds of motors. Some of them we know better, some of them not. And if I ask you, okay, please, in exam, what does the word molecular motors tell to you? Which names come to your mind? Then this could be a good starting point for us to talk and to see what we remembered, what we didn't remember from the course. Okay, so don't feel stressed. Everything is fine. I am getting some worried emails about exam preparation from mostly good students. You're all good students. Don't worry about this. So we'll be fine. You just attend classes, try to focus, you remember something, you forget something, totally normal, everything will be fine. Now, our last break, 10 minutes, and in the last part of today's class, we'll talk about methods used to probe molecular forces. And it will be not difficult. So for most of you, that will be kind of easy trip through different methods. You don't need to concentrate, you don't need to think about much, so it will be like a Hollywood film. Okay, so let's make a break, 10 minutes, see you in 10 minutes. we will make it quite easy for us. So please take it as a more or less relaxed work through the this group of methods applied recently for measuring cellular forces. The goal now is just to get an idea what is the spectrum of methods used for and applied for measuring cellular forces. What are the main principles that can be applied for that? And what's the range of forces we are dealing with? So that later, if you are in some talk with your colleagues and some mechanism is mentioned, some methods mentioned, you say, oh, I know it, I've heard about it. It has these and these, advantages or disadvantages. Or, for example, if you are asked about, so what could be used to measure such molecular or cellular forces? What are technical opportunities we have now that you also say, okay, I've heard about this method and that method. So this is the goal. You see, so it's not very ambitious goal. And but I hope still that you enjoy this finalizing part of today's class because there are some beautiful and very elegant ideas behind of it. Some keywords that I rather put listed for me, just not to forget about them, are listed here. We already spoken about Tensegrity model. I will tell you about some interesting aspects of Prp channels now, and then we will take a look what are advantages and opportunities, traction force microscopy, microbial respiration, flow chambers, atomic force microscopy, optical tweezers, and cell drum technology bring to us. So that for every this technology, you have certain ideas, certain image in your mind, and if needed, you can like bring certain information if needed. You know that cells, some mechanical aspects of cell functioning are very important in medicine. Just to name very few, in many labs, in our lab and also in Research Center Jülich, many labs are dealing with artificial skin constructs. So it's tissue engineering and making artificial skin. And if you just try to culture cells, for example, keratinocytes together with fibroblasts, you can get products that resemble human skill from cellular point of view, but most of these constructs will be not mechanically strong. So they cannot actually serve as actual skin substitutes because they are not strong mechanically. So there need to be more understanding and efforts to make them really usable. And one more example, not to make it very long, are cancer cells. You know that as tissue gets inflamed, gets inflammation or cancerous, transformation, it is usually getting more rigid. And on the level of the whole organ, it is usually started by palpation or elastography, but it shows itself also on the level of cancer cells. So as we are noticing that some tissue types or cell types are getting more stiff, more rigid, mostly this is a signal something goes wrong with them. Again, this can be used as a nice opportunity for diagnostics methods, measuring mechanical properties of single cells and tissues helps us in early diagnostic of cancerous states and other pathological states. Also, our cells are living in certain mechanical background environments, and they deal with mechanical stimuli every second of their life. And they have developed mechanisms that enable mechanosensation. So cells, they respond and generate mechanical forces. And here I wanted just to briefly show you some candidates for cellular force sensing systems. There are many publications, so some of them include ion channels that can open or close depending on the stress applied. As we'll see later, also tyrosine kinase receptors respond on stress. The G protein coupled receptors have many interesting mechanical sensing aspects. Caviola, the membrane invaginations many cells possess, can also perform mechanosensing function. The proteins in our inner ear, our auditory organs in the cochlear system, they also have mechanosensing, because without mechanosensing, you cannot sense acoustic waves, right? And some researchers also think that the Tensegrity ensembles you can find in our body, they also can be used, can be exploited for mechanosensation purposes. So as you see, there are lots of interesting mechanisms or devices our cells use to sense, to measure mechanical forces. Why? Because if the mechanical tension changes, the cell must react on it. It must change its growth pattern, it must rearrange its cytoskeleton, it must decide either it dies because the stress is too high, or it will divide in order to compensate stress, or it may change its shape to compensate the stress. So there are lots of important decisions the cell must meet depending on the environmental mechanical stress. Here are just a couple of few explanations. So one interesting example are so-called Prp channels. They are everywhere in our body. TRP stands for transient receptor potential. Transient receptor potential. So I put it in our chat window, transient receptor potential. These channels easily convert mechanical information into electrical signal because they let sodium ions in. And as you know, increased concentration of sodium in the cytoplasm means excitation. It is a depolarization of membrane that leads to an excitation, and this will be then forwarded to the brain. One classical example of that is, for example, if our urine bladder is full, because so it's increased in volume, so the urine collected in the urine bladder leads to extension of the wall of the urine bladder, and those channels sitting in the wall of the cells in the urine bladder, they feel this mechanical stress, so they're expanded, and those channels open, and they send signal to our neurons and to the brain that our urinary bladder is full. And I hope everybody understands how important proper functioning of this mechanism is, right? Also, cytoskeletal proteins like actin can be harnessed for mechanosensing, and here's one more mechanistical model which I will not go too into detail. We don't need it, but it just to show that there are hypothetical models where also actin, for example, is actively involved in mechanosensation. The external forces being sensed, they will cause numerous cellular responses, and that in turn will manifest on gene expression level that would affect the focal adhesions of the cells. Also, cell-to-cell junctions, that is the object of our next class, will be also affected here. So you see the external forces, depending on their nature and amplitude, they will launch a whole cascade of signaling events, which is a very sophisticated but very interesting aspect of cellular life. So one of these cascades is shown here. So very simplified, our shear stress sensing system is shown here. Of course, not for memorizing and not to frighten you, but just to show what we know already about shear stress sensing in endothelial cells. Also, those things are, of course, of critical importance for those who work on a cardiovascular problematic. You know, many people are dying worldwide on cardiovascular problems. And many of them appear due to wrong signaling here. So in fact, many atherosclerotic events or ischemic events, they appear on this level. And if we can understand and control these processes, it will absolutely critically contribute to the health and longevity of humankind. So this figure shows and illustrates what I already mentioned today, the fact that cells exert forces on the environment. And in this case, researchers placed, I think that was fibroblast, they placed it to some soft support, to silicon membrane. And as the cell started to function and to move, it produced some wrinkles on the substrate. And we could see these wrinkles. And these wrinkles definitely show that the cell does exert those tensile forces on the environment. So I hope with this figure I can convince you even more that cells do produce pulling forces on the extracellular environment. So I hope and I think all of us, we understand the diagnostical value and the importance of biomechanical measurements also on the, not only on organal level, which is clear, it's absolutely critical for cardiovascular, orthopedic, respiratory studies, but also on the cellular level. And with this motivation, I want us now to enter the methodological field of biomechanics, of microbiomechanics. By the way, also, if you're interested in coronavirus thematics, you probably heard about interesting aspects coronavirus causes in our lung, and the fact that many patients with coronavirus infection, they do not notice that they have infection. They can breathe normally, but their oxygenation level is dropping very significantly. Interestingly, this is related to the remaining elasticity of the lung tissue, And coronavirus, in contrast to many other pathological agents, does not change the elasticity of the lung tissue too much. So that's why patients with coronavirus, they do not feel that there is a problem in their lung. They do not feel any mechanical change in the resilience of the lung, and they say, okay, everything is fine. So the body doesn't send any signal, oh, something is wrong with breathing process. Everything is fine. But because of the lung damage, the oxygenation is falling down, and sooner or later, many of these patients, they need to get intubation and ventilation, artificial ventilation. So again, this biomechanical aspect appears here, right? So what are challenges of microbiomechanics? So of course, we deal with very little scales. We deal with very short events. The forces we measure here are mostly in piconewton scale. The cellular reactions are very sophisticated along the fact that it's a viscoelastic. That means we have to consider lots of viscose and elastic components in the biomechanics. It's a lot, including cellular reactions. That means compensational effects. So if cell reacts not as inert matter, it also actively reacts on what you are doing, right? And this makes it more complicated. If you take a piece of rubber, you can just check its properties, and if you do it now, or 5 minutes later, or 1 hour later, mostly you will find its properties same. But if you do it same with cell, the cell says, oh, what's going on here? I am seemingly subjected to some stress. Let me now do something against it, or let me deal with it. And what you will notice, there will be lots of compensatory reactions from the cell. And of course, this brings more troubles to your life. And the last point, which I called private life of cells, let me explain. You know, cells may be, which you are studying, they might be different cells at different phases of their cell cycle. Just think about if you take a young cell or old cell. What if you take some resting cell or the cell which is just going to divide? You will definitely find differences between them, right? And this is again one more unknown contributing factor, confounding factor, that makes your measurements hardly interpretable. So you can never know what the cell is now doing. You just take two identical cells, you measure them, and you find totally different results and say, what's going on here? And the reason was that one cell was going to die and another cell was going to divide, but you never know. So that's why it's difficult. So there are many interesting challenges related to micromechanics, and that's why it's, of course, an interesting field that requires high-tech things. So what's the range of the forces we're talking about? As I mentioned before, we are here in piconewton range. So starting with 1 piconewton, that usually characterizes protein-protein interactions. 60 piconewton is a typical range of forces involved in protein folding, unfolding. So if you want to unfold the protein, you need to apply this force approximately. Also, the same is true for DNA, double-strand DNA stabilizing forces. And of course, if we are talking about not hydrogen bonds, but about covalent bonds, then the forces involved are much bigger. In our course, Cellular Molecular Biophysics 1, we've spoken a lot about why we typically don't use piconewton, but rather kilojoule per mole. I don't want to repeat it now. All of you who will attend, who will take part in our Cellular Molecular Biophysics 1 course, will learn it with me together. Okay, and here we are seeing the nice range or huge range of the methods applied for measuring cellular and molecular forces. Let's take a couple of minutes to see what's going on here. This is obviously an atomic force microscopy approach. These two methods that resemble the traction force microscopy, which I mentioned today, but I will show more pictures right now. This picture indicates usage of micropipette systems and aspirational techniques. Here we are using some magnetic nanobits. There could be some paramagnetic particles that can be also applied for measuring cellular forces. This picture illustrates optical tweezers, also called laser tweezers. Here again, something close to atomic force microscopy. This picture illustrates low chamber approach, and in this case, I don't know how to call it. It's some mobile substrate. So we just place a cell between two supporting elements, and we just measure what is the force applied by the cell, or other way around, we can apply certain force in a controlled manner, and we can see what happens. If you try to write down what I just was telling about, sorry, I was too quick, but most of these methods we will now discuss in more details, and later you will easily assign any method, any name to all the methods we talked now. So now we will start our travel through these groups, taking a little bit more details and paying a little bit more attention to the methodology. If you later have any questions and if still somebody remains unclear, of course we can talk about this. So the first group I want to address, and now we're here in this cluster, let me label it so that we do not forget anything. Instructional module. Called traction force microscopy. The idea here is that we make our substrate flexible. Let me make some drawing here. Just a moment. So here I will put an empty space, and so now it should work. Right. So let's place a cell on a substrate, but let's make the substrate soft, so it will be soft. So let's use some soft material. Do you know some nice, suitable soft material? Just think about this. So maybe some sort of gel, maybe some hydrogel. You can use also polyacrylamide. You can use agarose. We can use, what do you know else? For the acrylic acid, you can use silicone, of course. Yeah, like gel. So some gel, like you can use also collagen, scaffold, so something soft. Okay, so something that in the range of several kilo Pascal Young's modulus. And of course, you also need some harder support. That could be glass, for example. So that would be hard. And this will be soft. Well, so now you already know what happens if a cell, if you place a cell to this support. We already know that cells produce pulling forces, right? So they will actually, they will pull the substrate so that sometimes even wrinkles can be formed. So in some extreme cases, you even can see wrinkles to appear on your substrate. But in our setup, maybe not, because we have here also hard background. But what you can see is, if you put some particles here, so let me now put some particles here. Then now, as the cell starts to apply forces, those particles, they will move, right? Because they will have displacement because of the forces produced by the cell. So, and now if you take a microscope, so this is your objective of your microscope. And now if you look through it, you will see that these parts have not any displacement. So it was at its initial position. But this part, for example, it moved, and you see it with microscope. For that, you will need to take two images. So first image you take before the cell is doing something, so initially, and then you let cell to exert forces, and you take second image. So then you have two images, and those two images you can apply, you can overlap, and then you see which points on your image have shifted, and this will give you a so-called traction map of your of the substrate. So you are basically tracking how particles in your soft medium moved after cell applied forces on them. By the way, the particles, they don't need to be applied in a very ordered manner. You can just mix them with gel and they can be totally disordered. It's no problem. You will have some pattern of their distribution at the beginning. And then a couple of days later, you can take yet another photo. And for every single particle, you will be able to see which particle moved in which position. And after you converted displacements of particles into pseudo color, you will get such a map which will indicate places where displacements of your gel matrix particles were maximal. And as you did it for this cell, you saw that this particular cell applies maximal forces in this and these directions. And here, actually, nothing happens. So here, nothing, right? So you see nothing changed. So this is the basic idea of the fractional force microscopy. We track displacements of matrix of substrate particles produced by the cell, by the forces from the cell. I hope that was enough for us just to get the idea, and more we don't need, okay? So just to get an idea, to see the principle, and let us go further. A second. So now let us speak about these methods. Here. Those are shear stress or shear force methods. Mostly we are dealing with flow of liquid, and liquid as it's passing by, it produces so-called shear flow shear stress, which many of you maybe know from your fluid dynamics course. And so, in fact, there are various options here how you produce this fluid flow. Either you can do it in by applying rotating cones, so you make liquid rotating exactly like you do for coffee cup. As you mix it, you make a rotation and the liquid moves, and it produces shear stress to the cells here. Or you can make a flow chamber, and many of these chambers we have in our lab. So you let fluid to flow through it, and by doing so, you can, in a controlled manner, you can produce shear force. There are some formulas that I'm not giving to you, how to calculate the shear force if you know the viscosity of liquid, the height of the chamber, and the flow speed. So you only need to know height of the chamber, flow speed, and viscosity, and then you get the shear force. Oh, it's too early for that, sorry. Now let's consider the group of atomic force methods. Now let me again move to this domain, and we are talking about, so that was 1, that was 2, and this will be our group number 3. Those are methods based on atomic force microscopy, AFM. Well, again, I am not sure, depending on your background, you may know a lot about atomic force microscopy. For example, in the laboratory from Professor Schöning, there is, I guess, some atomic force microscopy device, a compact version of it. But let me still briefly introduce the principles so that we understand the idea. So here we are probing the surface by using a cantilever, a special device which has some very sharp, very little tip, and we are probing it, and the displacement of the cantilever as it travels through the surface can be recorded and then processed to make a picture. And it carries the information about the profile of the surface, and as we'll see right now, also it can record the mechanical property of the surface. So basically, you are just measuring the displacement of the cantilever here. So in order to measure it precisely, your cantilever needs to be very small, of course. So typical dimensions are 100 micrometer in length and very sharp tip, and the area you are probing is quite little. So for example, in a couple of hours, you can only sample this little area. And even for this tiny spot, you will need a couple of hours. So also the tip needs to be made of very sharp and very robust material. So here's silicon nitride should be used. It's a very hard material, and usually doesn't break easily, and that's why we need to keep it as sharp as possible to address single molecular places. The atomic force microscopy can be applied in very different modes. So the very old mode is so-called contact mode, where you just drag it along the surface of your sample, just drag it, and as it meets any places that are elevated or deepened, you will see that it will follow the profile, and this you can just really record. But it has a disadvantage, because if you see some edges like what I'm showing now here, it will cause damage of your sample. So, you know, if you drag along the surface, you can damage it. That's not so good. That's why in later some non-contact mode will have been applied, where the distance between the tip and material is always not zero, so there is a gap between them. And later, even more advanced mode has been applied, which is so-called tapping mode. To me, it resembles as a blind man use its stick to probe the ground to check if there are any holes and obstacles. In the same manner, this blind man method can be used here, so tapping mode. So the cantilever continuously oscillates up, down, up, down, up, down, and it records the profile of the surface. The most advanced method not shown here is to use acoustic coupling method, where we make cantilever to vibrate at certain frequency, and depending on the forces acting between it and the surface, the amplitude and frequency of vibration will change. And this is what the most advanced devices use at the moment. So frequency changes caused by contact. Again, I'm doing it quite quickly just to give you some ideas. Okay, so I hope that you all now are a bit like relaxed, drinking something tasty, and not trying, you know, to write down hectically everything what I'm telling. Please just take a look at slides and look. It is really like Hollywood film. You don't need to record everything or write down everything. And if you have a good Hollywood film, then usually you will remember what was it, right? And this is our aim. Here are some examples of atomic force microscopy images: gold surface, bacterial plasmids. You see the images are quite nice, and even in the case of gold, we can even make images on single atoms of gold. So these bumps you see on the surface, these are single gold atoms, and those spheres you see, they are electron shells, which we probed, not we, but this group probes on the gold surface. So the resolution is really amazing. But what it does have to do with measuring forces. Well, it's quite easy. Look, we use the atomic force microscopy setup with cantilever to measure how strongly, for example, these two cells are attaching to each other. We make one cell to attach to the substrate. We can glue it, in fact. We can glue another cell to our tip of the cantilever. There are many methods how to glue them. And now, as we put them together and let them react, we can measure how strongly these two cells are holding to each other. So we can measure the adhesive strength between two cells. And this we can do in different time scale, quickly, slowly. This is, for example, one possible approach. You just start from some point away. Now you will approach them, and you brought them into contact. And then you try to detach them, and at this moment you're trying to detach them, you will measure some forces acting. And in fact here, depending on your height, on your altitude, the force will change, and this force as a function of altitude, you can then record, and it tells you about how strong is the force that acted between this red object and green object. If there are some colorblind persons among you, I'm sorry for the drawing. So the circle is red, and this structure is green. So I had twice already students in my group who were slightly colorblind. So that's why I'm mentioning. And that can be quite easily calibrated and used if you know the elasticity or spring constant of your cantilever. And if you measure the displacement, you can easily find the force simply by using Hooke's law. Quite easy. Yeah, thank you for this question, Lorena. So she's asking about, does it work for detachment, or also can it be used for compression in other way, so in the opposite way? And the answer is yes, certainly, exactly. And this is what the next slide will show to us. Thank you for this question. We can also use it in a compression way as well. And this is what very nice thing is actually. Look, we can push the cantilever, and we can measure how much is the resistance against the cantilever coming from the cell. And we can actually map, this is our cell, and the points, they show the places where we measured the stiffness. And then we can make a two-dimensional stiffness map of the cell. So if you make many measurements, you can say that this place was hard, for example, and this place was soft. So what appears as a result, it will be a two-dimensional mechanical mapping of a single cell. Really fancy, really nice. I hope you like this idea as well. And this is what is absolutely possible today. So we can measure every single location in the cell, and we can know if nucleus is more harder or softer as compared to the cytoplasm, for example. By the way, I want maybe immediately jump now to cancer cells. This is applicable for cancerous cells. This is what I mentioned before. And you can either prove the stiffness of the cells, and you will notice that cancer cells are always harder than healthy cells. They are more stiff. Or you can apply it in a profiling manner without measuring force, but rather measuring surface. And then you analyze the fractile nature of the surface. So what are irregularities of the surface? And also here it has been shown that cancer cells have different fractal geometry as compared to healthy cells. So in other words, the profile of their surface is different as compared to normal cell. And you see here on the histogram that by using atomic force microscopy, you can clearly distinguish between cancer cells and healthy cells. One other nice thing you can do with atomic force microscopy is, for example, to measure how firmly channels are sitting in a membrane. So you can take a channel protein and you can pull it out of the membrane, and you can measure the force, how strongly it holds in a membrane. Or you can prove how much force is required to unfold the protein. So you have a folded protein, and you take one end, another end, and you pull and you measure how much force is required to unfold a protein. Maybe pragmatically not so important, but funny. Okay, so atomic force microscopy is definitely one of the most powerful methods used so far. Now I'm moving backwards just to record our next success. So we did number 3, and now we have only a couple of other methods to discuss, and then we are ready. Now we are moving to these methods, and they are called optical tweezers or laser tweezers methods. That will be our 4th group. Optical tweezers methods. Here. The thing is that some of you know and some of you maybe not is that light or any electromagnetic wave can push, so it possesses momentum. Although photons have no mass, as they fly with speed of light, they get mass because of their motion. And they have momentum, and each photon has momentum. That means if photon bounces on some surface, it produces force in the surface, it pushes the surface. So that means every time you expose your face to the sun or your hands to the sun, at this moment sun pushes on your hand, but of course very slightly, so you don't notice. And this is, by the way, the reason why comet tails always show in the side opposite to the sun. So if you observe a comet which is orbiting the sun, the comet tail in most cases will show in the opposite direction. The reason is the pressure produced by sunlight. And by the way, some colleagues of mine, Professor Dachwald in Aachen, it's Faculty 3, Aerospace Engineering, they are developing spaceships that use the sun pressure to propagate rockets. So they are working on projects about sun sailing. So you make a huge sail, and the sail will catch the energy of the sun, and it will push your spaceship away from the sun. So sounds almost, fantastical, like a science fiction, but these are the projects our colleagues in Aachen are dealing with. What we are going to use is we want to use light as a sort of forceps, also called tweezers, pincetter. So we want to hold particles by using concentrated light. I think you know that the best concentrated light is a laser light, so that's why we will use laser light. And with the laser light, in fact, you can apply pushing forces on particles, so you can push them. Unfortunately, I'm not showing videos to you because they don't work in this system. I don't know why, but if you check in the internet on YouTube, you can find lots of funny videos showing optical tweezers. Let me just briefly say that light can push particles, but even more, if you play with the reflection coefficient of the particles, with their reflection number, you can make light to move particles to the left and to the right, because the resulting force will be not only pushing, but also shifting particles to the side. And these are quantum mechanical effects that can be used. What we only need to know is you can use focused, concentrated light beam to manipulate particles. You can move particles, and those particles are in nanometer scale, and you can use this particle to probe cells. This is the basic idea of the optical tweezers. So you use laser light, and you use usually polystyrene particles, and you apply these particles to poke, to probe cells. So these are particles, and this is how you manipulate it with light. Blah, blah, blah. I will skip it. Here's the setup. So you need two laser beams just to compensate each other. Here, place your specimen. Here, one laser beam is coming. Here, another laser beam is coming. It's done just to compensate the pushing force. Otherwise, you will push your particle away. And that's why you need to push from both sides so that the particle will remain in the focal plane and will not fly away. But now you have two hands from the left and from the right. Every laser beam is a hand, and now you catch your particle, and now you can move up and down. And by doing so, you can now exert forces on cells and molecules. Again, it sounds quite, unrealistic, but this is a real device. Unfortunately, we don't have it in the lab because it was a bit expensive for us. Maybe someday I dream we can buy such a device, and I could show it to you live. So optical tweezers, for example, can help you to catch one single enzyme, for example, RNA polymerase, and you can measure forces between RNA polymerase and DNA. Or again, you can catch some protein, and you can measure how big force is applied if a protein is folding, something like this. Now there are even high throughput systems that not use just simple laser beam, but also elliptically polarized, circularly polarized laser beams. And there are also high throughput traps where you can produce many laser beams, and you can trap many particles. So you have like array of particles that are trapped by laser light. Well, I think it's enough for laser tweezers, and the two remaining techniques are coming now, and the next one will be this one, number 5, and then we'll talk about number 6, this one, and then we're ready. So about magnetic particles we will not talk. I don't have anything to tell you. Okay, now 5 and 6, and we are ready. Okay, so please 5 minutes patience, and that will be it. So microbial respiration. In this case, we're using very thin nano capillaries, micropipettes. We cannot make them really small, so we have to. So mostly they are 1 micrometer in size, sometimes 3 micrometer. And here you see how we can catch a red blood cell, and we can examine its stiffness. So for example, if you smoke a cigarette, your red blood cells will be more stiff, and we can catch them with micropipettes, and we can measure how much sucking pressure you need to apply to deform your red blood cell. And if you smoke a cigarette, we will immediately notice that we need more sucking pressure, and we will tell you, oh, you just smoked a cigarette. I can see it. Well, but if you smoke a cigarette and if you eat chocolate afterwards, then we cannot see it because chocolate will compensate for cigarette smoking. So if somebody smokes a cigarette, it would be a good idea to eat a chocolate afterwards, because they sort of eliminate each other, at least biomechanically. So these are some figures from our lab. We have a micro aspiration setup in the lab, and we use it. So here are some pictures. Of course, such setup can be also used for micro-fertilization. So this is not my photo, but this is where artificial cloning experiments are done. So you can use same setup for cloning experiments, but we don't. And here we mostly use red blood cells, and we check their rigidity, and we study their mechanical properties. Before we do the very last chapter, I want to tell you some funny stuff. We had a student from Poland. She studied the forces exerted by white blood cells. And what we did, we used micropipettes to catch leukocytes. So we found a leukocyte in our blood, and we held it very firmly, and then we put some bacteria. I maybe draw it like this. So that was the leukocyte, that was the micropipette, and we produce some pressure which will hold it, and here we place some bacteria. And because now bacteria produce substances that are attractive for white blood cells, this leukocyte tried to move towards bacteria to catch it. So our usual immune reaction, you know, just to catch it. And we trapped it, but we holded it. And it was really nice to see how this white blood cell tries to catch bacteria. So it was really trying to crawl in this direction. So it fighted and fighted, but we didn't allow it to crawl. And we measured how much force you need to apply to hold leukocytes. So we measured the force applied by leukocytes. It was quite funny, and later she found her place in working place position in France, and made her PhD, and also published a Nature paper. So that was really good scientific career. But what she did in our lab was this experiment. Here are some questions. Let me ask them. Yes, so Saskia refers that she needs to go to work. As I understand, actually we are ready. So sure, no problem. You can leave. It's just very little thing that we need to discuss, a couple of minutes. But you can go surely. It's not very important. What we will discuss. Then there was a question: can you damage the cells? Well, it happens indeed if the edges are too sharp. That red blood cells they burst, but mostly due to fabrication of microcapillaries, they have quite smooth edges, and that's why it usually doesn't occur. So this method is relatively gentle to the cells. So if you notice that you did something wrong, you can take another capillary, and then it mostly works. So it's not very much hurting the cells. If the force is greater than cell structure, well, the question was if the force is greater than cell structure. Oh, you mean, yes, so if you apply too much force, that the cell can be damaged. What we mostly observe, if you apply too much force, the cell will enter the capillary and will pass into the system. Or it can be just broken indeed, so and you will notice it immediately. If you apply too much sucking pressure, then the cell just breaks, and it's like sort of just falls apart, and you say, okay, sorry, we killed you. Yeah, it happens. But of course, with a special pump, we can very exactly control the sucking pressure, and mostly does not happen. And our last method, which will finalize today's class, thank you again for your patience. We will be ready now. Is the group of methods where we apply tensile forces on the support, and one of the systems I want to present because this is a system which has been developed in Jülich. And of course, we are pretty proud of it. It was our group. It wasn't me, but I participated. And there was Professor Gerhard Hartmann, Professor Jürgen Cevik, he is now professor in Hamm, Dr. Peter Linder, and Dr. Gossmann. So this is the working group who worked a lot on the development of such system. And by doing so, we are able to measure cellular forces for cellular monolayers. So this is the technology that is patented, and it belongs to Jülich, to our lab. So that's our know-how. It is a silicon membrane, and on the top of the silicon membrane, you have a monolayer of cells. And this system is called cell drum. So now, if you put the cell drum, it will be here. Let me draw. Here. No, sorry. I'm wrong. Here. If you put your cell drum here, and if you apply some sucking pressure or pressing or pressure in one or other direction, you will be able to produce displacement of the membrane. If you push, it will be like this. If you apply aspiration, it will be like this. And so you can move the membrane so it will bend. And if you apply controlled pressure, you can measure how stiff is your membrane and how stiff are your cells. So by checking the bending, the curvature of the membrane, we can measure how stiff the cellular monolayer is. I think it's just an animation. I can try to apply it. It didn't work. It works a little bit. Yeah. Oh, yeah, it's not so important. So, and this technique has many advantages as compared to the classical techniques. So this one is used in Research Center Jülich by the group of Professor Merkel and Professor Hochmann. And they are our competitors, so to say. And this is their group in Forschungszentrum Jülich, and this is our setup. And you can see that according to a finite element analysis, the loads we apply is more uniform. So we can make it in a more controlled way, and it's more advanced. And even two guys from our lab, Peter Linder and Matthias Gossmann, they had a startup. They established a company, Innovitro, which has headquarters in Jülich, and they are quite successful. They have now very many projects from pharmacological companies because they use the setup to measure cellular forces, and they earn lots of money at the moment. And I'm of course very proud and very happy that our students who worked on cellular forces now were so successful in their business. You can visit their webpage. They sometimes offer master positions. They're called Innovitro. And they have very beautiful webpage. Okay, because of time reasons, I can skip these figures. They are not so important for us. And in fact, we are ready with our overview of the cellular forces. Here, just to finalize, these are main methods we acquainted today. And here I try to put the range of the forces you can measure. All of them is shown in piconewton range. Again, I'm sorry for making it maybe a bit long today and maybe a bit fast today, but I still do hope that you got a certain idea about what kind of methods are used nowadays to probe cellular forces. So you have several pictures in your mind. So you've learned a little bit something about what can be done, what cannot be done with some methods. Some of them are more rough and some of them more fine. Some of them, they cost more money and some less. But I think we still made a lot today. And now I think it's time to wish you a nice weekend. And thank you again for your attention. So next time we meet, next week, we will talk about adhesion and junction structures. So please find and download our next script from Ilias, which is called Adhesion and Junctions. This is number 2. So we will talk about adhesion systems in the cells. So thank you very much. Have a nice weekend. Have a great next week until we meet next time. See you. Bye-bye.